Hello everyone, I'm Hafumi Nishi from Tohoku University. In this video, I'm going to explain our research about genetic variation on human SLC transporters. Membrane transporters are proteins that mediate the transportation of various molecules like ions and sugars across membranes. And SLC transporters, solute carrier transporters, are known as one of the most prominent protein families in the human genome. And they are also known as a potential therapeutic target. Here, we developed an integrated SLC protein database called CastleDB to provide biological and biochemical information of all human SLC transporters. CastleDB includes various kinds of information such as sequence, structure, pathway, genetic variant, and biological networks. The information were extracted from these databases. Here, the screenshots of the top page and the detailed page of CastleDB. You can access CastleDB from this URL. As a part of database construction, we mapped all available genetic variant in gnomAD database to human SLC transporters. And we also performed the detailed analysis of genetic variants based on the 5 regions of transmembrane proteins. The definition of 5 regions are shown here. So usually we use 3 regions to define the different location of transmembrane proteins, like extracellular, transmembrane, and intracellular. And here, we employ the definition of 5 regions taken from Higuchi et al. That includes 2 unique regions. These are the marginal regions between extracellular and transmembrane, or transmembrane and intracellular. And the transmembrane regions are predicted by TopConf server. Here are the results. We divided the genetic variant into different categories based on the minor allele frequency, like intermediate or common. And as you can see, even for all variant, there's some tendency that transmembrane region has less genetic variant compared to extracellular or intracellular. And this trend becomes prominent in intermediate variant or common variant. And here we also created a new category called negative score variant. And the meaning of negative is the substitution pattern of the genetic variant are scored negative in gnomAD substitution matrix. So this variant implies that there are harmful mutation for proteins. And it is clear that the transmembrane region, RC region, has less harmful or less common variant. And interestingly, when we focused on the R2 and R4 regions, for common variant and negative variant, there are differences between the R2 and R4, even though the R2 and R4 are the marginal region between the transmembrane and extracellular or intracellular regions. We further calculate the residue mutability of each region. So residue mutability is defined as the ratio of frequency of each amino acid between variant and all residues in that region. So basically, if the value is more than 1, it means that amino acid can be found more frequently among genetic variant. And we found that arginine has the highest mutability among all 20 amino acids. But however, the value greatly vary among topological regions for common variant. So in all three regions, we did not find any arginine mutations. In contrast, for valine, it also has a high mutability, but the high mutability was observed in all three regions, and there are no common variants in all four regions. And we also confirmed that the residue mutability were almost unchanged when we include all variants. In summary, we mapped all available human genetic variation to human SLC transporters and found that variant with a different minor allele frequency distributed differently in terms of the 5 topological regions. For example, transmembrane R3 regions do not have many genetic variants, and also the R2 and R4 are both marginal regions but have a different trend. And also the residue mutability of each amino acid showed unique characteristics of each amino acid. For example, arginine mutation were avoided in transmembrane regions, while valine mutations were abundant in the transmembrane regions. And our SLC database is available at this URL. Thank you for listening.